Okay, welcome
Perfect.
everybody. My name is Juan Viguera. I am the Chief Marketing Officer for Veravanti. Veravanti has been around for almost 10 years now. This project started by a research group at the University of Washington. And so all the IP that was developed by Eric Seibel and the group at the university is now controlled by us, and we're moving the ball forward. And we hope you're going to enjoy this presentation. This presentation is being recorded. We have people from around the world joining us. As I mentioned before, we have Europeans, South Americans, Southeast Asia, and it will be watched by people from Japan and Australia later on today. Most of the information that we're going to be providing today is somewhat confidential. It's also, we're going to make some statements that are forward-looking, so we need to legally tell you in advance those two things. Who's going to present today? Joe McMorrow, who's our founder and CEO of the company. He may or may not be present. I'm waiting for him to join us, but he is the founder. He was also the founder of Verathon, a company that he sold a few years ago for about $300 million. He's used about 20 million of that money to invest in Veravanti where we are today. So it's a great inventor. It's an engineer, one of the best engineers I've met in my life. He's an Army veteran. He went to Vietnam for fighting for the US, and he's built several businesses. Most of the presentation will be handled by Luis Savastano. He's a doctor, a well-renowned doctor. He works right now out of UCSF in San Francisco, and he'll explain all about himself. As I mentioned, I am a marketing officer. I've been around the medtech industry for about 5 years. I used to work at Microsoft. I spent 10 years at Microsoft, retired from Microsoft in '99, and then since then I've been doing investments around the world. So we think that we can help you understand this technology pretty well. Why Veravanti? We have invested about 40 million so far in this technology. Trust me, it's quite expensive. It takes a lot of time to get to where we are, but you'll be impressed by the quality of the videos you're going to see today. We recently closed a Series A financing round for about $31.5 million, giving us a valuation of about $120 million US. And as I mentioned before, Gerald, our founder, has been the one that has funded the company for the most part until today. The good thing is that we've learned that our technology is going to disrupt the market. We think we have potential to make a huge difference on patient healthcare in the future. What we know today is that about 8.1 million angiographies happen in the US alone. And we also know that about 30% of the CTOs, they fail for many, many reasons. But the most significant reason for failure is because doctors cannot see very well. So they decide to just send the patient to have an open heart surgery. So that's the number that creates a lot of anxiety for us to make sure that our product gets to market as soon as possible, because we think we can help a lot of those patients that are today going through CTOs. In terms of why we think this technology could be beneficial to all of us is because we see the existing technology, we see what IVUS does, we see what OCT does, and we see some of the key limitations. We believe that their image quality drops as the diameter of the scope gets smaller and smaller. We also believe that the contrast media that is sent creates vessel shadows, and the shadows are difficult to understand. And Luis will spend quite a bit of time on this, so I just want to quickly jump through this. But again, the CTO guidance fails up to 30% of the time. That to me is a number that creates a huge problem for the numbers in the US. There's about 500,000 cases in the US of CTOs, and 10 to 30% of them fail. And in some cases, a patient has to come back to the hospital 2 or 3 times. That generates a cost of about $47,000 per CTO procedure. And if you do the math, you end up with about $3.2 billion of losses created by this problem. So we hope with our technology, we'll be able to minimize this target market. This is going to be our first market we're going to go after. What you're going to see is that the SFE, the scan fiber endoscope, it's a platform technology. So we can apply it across multiple applications, from pulmonology to intravascular to urology and even dental. And Luis will spend some time explaining how this technology works in more detail. The real reaction to what we're proposing to the market today is what we're seeing out of LinkedIn. I post every Tuesday and every Friday on LinkedIn. And so far in the last 3 months, in 3 viral posts, I've gotten about 300,000 impressions, about 3,000 new followers into my profile. And most of those, out of those 3,000, are interventionalists. And so I'm having a lot of good discussions with them about where we are and where we need to go. And some of the feedback you see here is from doctors that are in this business. And I want you to focus on the one on the bottom left from the European CTO Club. This is a club of doctors that have at least 1,000 CTOs performed. And so they're pretty much experts. And we're going to be with them in Rotterdam on September 11 to 12 for their annual meeting. With that, I'm going to pass the baton to Luis. This is a picture from last year when he presented at TCT in Washington, D.C. We will also be at TCT in San Francisco later this year. And Luis will take you through the details of what this technology is all about. Luis.
Thank you, Juan. I will bring my slides up and then we can talk about, as Juan mentioned, I am a vascular neurosurgeon, trained at the University of Michigan, then faculty at the Mayo Clinic in Minnesota, and most recently at the University of California in San Francisco. And I'm a basically full-time physician, so I have had the opportunity to experience many of the issues and unmet clinical needs, as many of the people connected in the webinar. And I've been lucky to be able to use the system under development by Veravanti for a few years now, doing the preclinical work, which is necessary before going to the clinics and start doing patients. So the goal for today was mostly to give you an overview of the technology and what are the things that we are seeing in our arteries that could, I would say, radically change how we think about cardiovascular conditions and how we treat them. So that's the goal. And the presentation is going to be maybe half an hour long. So we're going to have 20 minutes at the end just to have an open discussion and to answer any questions that you may have. So I like to start from the beginning typically when I talk about this, which is if you think about this, X-rays in the medical field have been around for about 140 years. And these one or probably the first medical X-rays that we have documented. The first time X-rays were used to look at cardiovascular conditions was actually in the 1930s by Egas Muniz in Portugal that developed a technique to directly inject contrast inside the lumen of the artery, in this case the carotid, on a patient that presented with a stroke. You can see how there is an occlusion of the carotid artery, and then there is no intracranial flow. And moving forward almost 100 years, we are still doing more or less the same with probably more sophisticated tools, but same concept, which is going intravascularly with wires and catheters, and then inject contrast and see where the contrast is flowing and try to identify based on the shadows caused from the X-rays, where may be the problem based on the presence of narrowing or the presence of an occlusion. And we do millions of these procedures per year. I think it's remarkable what we can do with this, but we would all agree that this is not sufficient to really understand everything that's happening inside our vessels. And that was the motivation of developing a new generation of tools that we put in a bucket called intravascular imaging. And there are two options right now that we use in our patients. What we're seeing on the right is based on ultrasound or intravascular ultrasound, which has the advantage that we can see through blood because it's based on sound on wavelengths. but the images are very grainy. It's really hard to interpret what we're seeing. And what we have had more recently, probably for the last 20 years or so, is what we call optical coherence tomography, which is based on light and scattering. We need to flush the blood to be able to basically see the wall of the vessels, but the resolution is better. Having said that, there are many artifacts. It's hard to really understand what we are seeing many times, especially if there are artifacts in the pictures. And at the end of the day, we're still relying on X-rays to do most of our procedures, to guide the wires or to deploy the stents. So in a way, these intravascular imagings, they help, but they have several limitations regarding the interpretation and regarding the fact that they can only see sideways. So in a way, if you're driving through a tunnel and the tunnel are our vessels, you don't want to be looking sideways. You want to be looking in front of you. So you know where you are going. You can guide your procedures. And at the same time, you can inspect better what is in front of you without the need of crossing something. So, you know, what I found many years ago, that what we actually needed is an endoscope. And an endoscope is basically a camera that you can put inside a cavity or a tube in the body. And endoscopes have thrived head to toe throughout medicine, but not in the cardiovascular field. And if you look at this, endoscopes for arteries is not a new concept, actually. There is actually a society in Japan that they cluster a group of physicians that they do angioscopy routinely. And what you're seeing here in the screen is a technology that was developed in the 1980s. The technology is based on coherent optical fibers, which means that you take multiple fibers and then you put them together and then you illuminate and then you capture back the reflection of the light. And what you're capturing on one side, it's transmitted through the optical fibers to the other side. And the problem is that each fiber adds diameter and stiffness to the device. So you are forced to use really small bundles of 10, 20, 30,000 fibers at most. And each fiber creates a pixel. So when you look at the images are dark, and you can see how they are very low resolution, and you can even see all the pixelation from the individual fibers. And that was one of the main reasons, despite being around for 20 years, this technology has not really taken over. So that's basically where we are. And what Veravanti has done is to develop a new type of imaging platform in the way that you have ultrasound and you have X-rays and you have MRIs. What Veravanti has done is developing a scanning engine for lasers. And the way this works is they took a single optical fiber and developed a tiny micro machine that fits within a millimeter catheter, within a 3 French catheter. And that machine is actually a scanner that is spinning or scanning the optical fiber, which transmits a laser. And the laser is scanning in front of the device. And what you are seeing here is the concept of having the laser beam scanning from the center and basically following bigger and bigger circles. And the light is projected forward. And then everything that is reflected is captured back by another fiber that goes to a detector and a computer that reconstruct images. And all this cycle that you are seeing here from the center all the way to the periphery, it's happening 30 times per second, which means that every single second, we are illuminating the field of view 30 times. and 30 photos are generated, which is more than enough to create videos. So this is another way to see what I was explaining, which is this mini scanner inside the tip of a 3 French catheter or 1 millimeter catheter. And you can see how the laser goes through tiny lenses, so can adjust for the focal depth and illuminate forward. And what you are seeing here is the spiral path I was explaining before. And this spiral path is formed by the laser beam. And you can use multiple lasers through the same single optical fiber. In this case, what you are seeing is an overlap between a red, a green, and a blue lasers, which are the 3 colors processed by our retina. And by doing that, you can create not just videos, but full color videos. And the resolution on the screen, basically the amount of pixels, depends on the size of the laser beam, which it results in over 1 million pixels in the screen. So very small size, extremely high resolution, and we are delivering just a small amount of energy at every single millisecond, which makes the camera to be cool. So there is no problem with heating. The camera looks actually like a cathode from the outside, but from the inside, it's pretty sophisticated, and it's formed by this very special actuator. And the catheter is single-use, disposable, and then it's connected into this machine, which is actually where the laser sources are, and the images are reconstructed. So what I'm showing you is what the technology shows you. In this case, is a human cadaveric model where you can see all the arteries, all the branches from the vessel. You are seeing 360 degrees. You are seeing full focus and full illumination. So what we have been doing for a few years is to developing the endovascular techniques to use this device in basically in vivo in animals. And then we have looked a few hundred surgical specimens and cadaveric specimens to understand how diseased vessels they look like with endoscopy. And what I'm going to show you is first how this looks basically in vivo. These are animal models, typically large animal models, and the device is inside a catheter. And you can see that just by looking at the tip of the catheter, you see the blood flowing over there, and then you can see the surface of the vessel. And then if you use a bigger catheter or a catheter with a tiny balloon, so you can stop the flow of blood, then you can flush the blood away with saline or contrast, similar to OCT. And then that's the view that you obtain within the vessels. And then what you can do is just to keep the balloon open, and then you can explore long distances of the vessel with the endoscope. In this case, we are pulling back, but at the same time, you can advance over a wire. And what you're seeing there is basically a young, healthy animal. And the animal can be, there are experimental models to develop atherosclerosis in the animals, and that's what you're seeing over there. What you're seeing are early atheromas that are coating most of the vessel, and this is the normal surface. So you can clearly start identifying early lesions of atherosclerosis. And then you can disrupt those lesions. And then you can start seeing clots, for example, like you are seeing here. And then you can start seeing dissections, like you can see there, like intimal flaps. And then clearly you can see rupture plaque with intraluminal clot. And then when we're talking about clots, this case is basically disrupted atheroma with thrombus, is a thromboatheroma. So what you start noticing is the development of the clot, which is attached, anchored to the surface of the vessel by these fibrin webs. And that is a specific type of thrombosis, which is different to an embolic occlusion, which is the case. What you can see here is one clot that migrated and has completely occluded the artery. And you can even see the color of this clot is white. So this is a fibrin and platelet-rich thrombus, which is different to this type of clot, which is a red, basically red blood cells rich clot. And then finally, you all know that not all occlusions are the same. And what you're seeing here is a dissection flap. And you're seeing this in front of you. You can clearly see that there is a pseudo lumen on the right side, and there is a true lumen on the left side. And by being able to detect which is the true lumen, you can actually enter the true lumen and push the false lumen away, which is critical when we're doing, for example, intentional dissections in chronic total occlusions to then find the true lumen on the other side. Those are some examples of basically in vivo images, but we are looking in front of us. And when we're looking in front of us, we can guide our procedures without x-rays, but with direct endoscopic visualization. And this is, again, in vivo. This is an O14 wire that you can see how we're using to catheterize this tiny branch. And then these are our two common procedures that we do, at least as endovascular neurosurgeons, which is thrombectomy for stroke. And you can see here that there is a thrombus in the lumen of the vessel that we have engaged with a stent retriever. And now we are pulling the stent retriever out. And in the process, we are dragging the clot with us. And then what you can see here on the right is another common procedure that we do, which is a coiling of aneurysms. And in this case, we're inside the artery, we're looking at the neck of the aneurysm, which is here on top. And then under direct visualization, we are deploying the coils. And we want the coils to remain inside the aneurysm and not to herniate into the parent vessel. And that is something that can be done directly with this endoscopic view. And these videos that you are seeing, actually, they are published and is work that Patrick McVeigh has done, where you can clearly see in vivo procedures where rather than relying on x-rays, we're doing this endoscopically. In these cases, we are using balloon guide catheters to flush the blood away, and this can be done. But in addition, as I mentioned, the technology is able to scan different lasers, including lasers that can travel through blood. And that is something that is in the research pipeline of using very special near-infrared lasers that have the capacity to travel through blood, to see through blood. And what you are seeing here is the renal artery of an animal. You can see that there are coils. And then using these near-infrared lasers, we can advance our catheter inside the vessel. And then you can see the coils and clots which are forming. Still grayscale, but seen through blood, which, as you can imagine, that could not just change the game for the peripheral vasculature, but as well for cardiac procedures, if you can have endoscopic view of the valves during procedures. So what I'm going to do next is just to show a few examples of phenotypes of ruptured plaques that we are seeing in our preclinical experience and how that could impact our clinical practice. One of the things that we're working on is trying to understand how the technology could help us to prognosticate the future of a plaque, basically to detect high-risk features and understand if a certain lesion is symptomatic or not, or basically if it is an incidental finding. And this is a common examples that we see in our practice. And imagine that you have an elderly lady that had a recent GI hemorrhage. So in theory, there is contraindication to anticoagulation and antiplatelets, that wakes up at 2 a.m. With difficulties talking. And upon arrival to the ED, she's back to normal. The head CT and the brain MRI looks completely normal, although the CT angiogram shows an 80% stenosis in the internal carotid. So the question is what to do with this patient. Are these symptoms a TIA that is caused by a symptomatic plaque that we should revascularize, or we should do medical therapy with antiplatelets and anticoagulation, despite the fact that the patient had a contraindication given the recent GI hemorrhage? Or maybe this is a benign calcification in an atheroma that is completely incidental, and we don't have to do anything. In this lady that just had 10 minutes of difficulty talking at 2 a.m., maybe she was just confused. And right now we cannot really, we need more data points to do decisions. And what I'm going to show you are two different plaques calcified that cause 80% stenosis, and they look exactly the same in the CTA. But if you look with angioscopy, you can see that this atheroma is just pure calcium. It's very smooth, and there is nothing actually concerning in the surface compared to this other one. You can see that this other one is as well eccentric calcification. You can see the calcium over there. But on top of the calcium, you can see that there is a clot that is sitting in the surface and very likely is at risk of causing another TIA or a major stroke. So the way we would treat this patient is very different based on the phenotype that we can see with angioscopy. The other thing that we don't talk often at all because we cannot see it is how old is a lesion, basically the chronicity of a lesion. We typically talk about a dissection, an ulcer, a stenosis, but we don't talk clinically, at least, about if this is an acute ulcer or a chronic healed ulcer. And this is another example. Imagine that you have this patient, 62 years old, that has, let's say that has another TIA. And then you do a CT angiogram, and then you can say, yes, there is about 50% narrowing, and there is an ulceration. But in addition, the patient has untreated AFib. So what do? You treat the carotid, you treat the AFib, you anticoagulate, you treat both. It's hard clinically right now to just decide. And if you're able to see, imagine this ulceration, not all ulcers are alike. What you are seeing here are two holes in the plaque, or actually ulcers or excavation, which are acute. You can see that there is necrotic material, which is this yellowish material mixed with clots. And then you can even see how it's like cottage cheese, is very friable. And it's very likely that the blood flow is going to keep washing downstream those, basically the necrotic material causing more TIAs and stroke, compared to this other example. This other example, you can see there is the external carotid on the top. You have the internal carotid in the center, and then you have a very large ulcer, but is mostly healed at this point. And is important because if this is healed and chronic, is unlikely to be causing the most recent symptoms. So these are just some examples of the structures that we're seeing. Now, so I showed you these pictures before, which is examples of coronary, cerebral, and peripheral vessels, which are narrowed or stenosed or occluded. So the next question is, what is the tissue occluding or narrowing the vessel? is calcium, is clot, is a plaque hemorrhage, is a dissection. It's really hard to tell. And unfortunately, it's not easy to see even with IVUS and OCT, especially if there is a complete occlusion of the vessel, because we cannot look forward. And this is very important, not just in the brain, but in the coronary as well. You're probably all familiar with basically this idea that there are cases of coronary syndromes that are likely to have a high clot burden. And based on the angiographic appearance, we're trying to infer how much thrombus there is actually in the vessel. And based on that, there are basically suggestions about what we should be doing depending on the thrombus burden. But no, I think if we are being objective, there is a lot of speculations when we're looking at this, and we can say, oh, this is actually thrombus versus a plaque or a thrombotheroma or something else. And if we have the capacity to see inside, this is how an acute thrombus would look like. This is a thromboatheroma, a fresh thromboatheroma. And you can see how the surface of the vessel is full of red clot, and they have these chunks of platelets and fibrin aggregates on top. And I know that this is an acute thrombus because this is an endarterectomy specimen. Basically, is a tissue that we remove from the carotid of a patient that had a stroke less than 24 hours ago. So this is something that has recently evolved. Compared to this that you're looking on the right, this is a specimen that was obtained in a patient that had a stroke more than 10 days ago. And then if you look at this, you can see that the phenotype of the thrombus is very different. You don't see that red color anymore. What you can see here is how there is a clot, and the clot is being covered by this fibrin network, and it's becoming in a way stiffer and stronger and more attached to the vessel. And I think it's important because it's not just the presence or absence of a clot, is as well what type of clot we are talking. This thrombus here is likely easier to be removed by suction thrombectomy, basically by aspirating the clot, and then is going to be more likely to respond to thrombolytics medications, compared to this clot here, which is basically organizing and is less likely to be aspirated and less likely to respond to medication. Maybe what we have to do here is angioplasty or stenting. And the same happens here. What you're seeing here is basically a critical stenosis caused by an atheroma, and then you can see how there is a tiny channel left of the true lumen. And when you're looking inside, this is what you are seeing. You see that there is some red, which implies that there is a clot, but the clot is not within the lumen of the vessel. The clot is actually inside the wall of the vessel, is inside the atheroma, is what we call an intraplaque hemorrhage, which is important because in this case, if you think about that, there is no point of using suction thrombectomy because the clot is under basically this fibrous tissue. And there is probably no point as well to using thrombolytics. There's nothing to dissolve. What is important is if you keep looking up, you're going to see that there is a big hole. That is the ulcer here that you are seeing in this angiogram. And then when you are trying to cross this lesion to revascularize, you all know that it's critical not to plunge through the ulcer because that can destroy, can damage the plaque even further and create more thromboemboli. So it's very important to find the actual lumen, the true lumen that is critically stenosed, which sometimes can be very challenging, especially if the inlet of the true lumen is very small and irregular. But now imagine that you are having this visualization in front of you, and then without x-rays, you can look down, you can see that there is a tiny hole, and that is the inlet of the true lumen that you can just wire under direct endoscopic view.
Luis,
So,
Luis,
yes.
this will be a good time. There's a couple of questions that I
Perfect.
cannot answer that you probably can. Marion has a question. Marion, can you join us and ask your question? Marion? So what Marion asked is that.
Yeah, just a moment. Hi, hello. This is just I think it's something that it's changed the game. So it's absolutely fantastic to see, for example, this intraplaque hematoma, and if you need to do aspiration or not, it's something that changed your mind absolutely. My question is about CTO procedures in the coronary. If you can do it in 7 French or 8 French, so if you're using SFA, you put it on one wire, and after that you put the second wire, CTO wire, on the microcatheter. You can do it in 7 or 8 French?
Yes, Marine. So the technology itself is going to be 3.3 French, which is 044, and that has a monorail system to put in 014 wire. So you typically what we use is a 6 French catheter, and with that we have plenty of space to have our system and a microcatheter next to it. But if you need more kind of real space, then you can use the 7 French.
Yeah. So you can do it in 7 French without problem.
Yes.
Oh, that's great.
The next question is from Joel Giblet from France.
Oh, I'm from the UK, but
I'm sorry, Joel.
I'll take the upgrade to being French. That's fine. So my question really was, so if you're doing this in the coronaries, in order to get this quality of picture to help you, you're occluding the coronary upstream with a coronary balloon. Is that a sort of over the wire balloon so that you can, because presumably once you've occluded the coronary, you're going to have difficulty manipulating wires past these balloons. So are you sort of having to preset with a microcatheter on one wire and over the wire balloon, and then a third wire with the SFE on, in order to actually utilize it to its full extent? Or am I misunderstanding how the technology is being deployed in the sort of real world?
Yeah, so we haven't started the clinical experience yet, and that is something that the procedural development still needs to happen in patients. I can tell you about the procedural that we're doing in large animals.
Okay.
And there are 2 techniques. One that is what we call the occlusive, when you have your guide catheter has a small balloon that you can inflate, and then you can advance your system over the wire. Or the other option is when you're injecting saline or contrast, the way that you do OCT currently, you acquire your images. And then keep in mind that you're looking forward and the depth of view is about 5 to 6 centimeters. So you just need 1 second to be able to look 5 centimeters down the artery.
Okay.
And again, I'm a vascular neurosurgeon, so again, be gentle because I'm going to show you coronary images. But the idea is this is swine. That in this case we have a 6 French catheter when injecting contrast, and then during the contrast injection we are obtaining these images. So there is this particular prototype did not have a monorail system, was not over the wire.
Okay.
So we anticipate that when we have the wire, it's going to be more stable and centered in the vessel.
Yeah, because presumably the 1 second for the contrast will be fine if all you want is the pictures, but if you want the real-time guidance to aim for the true lumen of a dissection or something like that, it's going to have to be quite carefully thought through how you're managing all of these things.
These cases, for example, this is live as well, and we have the balloon guide catheter. So we're opening the balloon, and then we can work for basically long time with this. Especially if there is already like a chronic total occlusion, that allows you to basically, you have an occlusion on one side, you open a balloon on the other, and that gives you a region without blood flow where you can work for a long period of time.
Yep, and it doesn't matter for the patient because there's a CTO further down the vessel.
Exactly right. Exactly right. Having said that, we realize that would be great if in the near future we can just see through blood, like literally navigate all the way without X-rays or minimizing the X-rays and do procedures. And this is the technology that I was showing you. Again, this is not true color. This employs near infrared, so it's going to be grayscale. But you can see here the artery, and then down the artery, you're going to see the coil mass, which is this. And in this case, the visualization is through blood. So it still needs work to increase the resolution, but we think that there is a clear feasibility path to have a platform that is true color, where you basically flush the blood away, and then have another platform that is able to see through blood, maybe at lower resolution and not just in grayscale, but despite that, you can clearly see the metal of the coils, and you can see the metal of the stents, for example. So you could do like complex Y stenting cases.
Look, Luis, there's one more question from Lucas from Poland.
Hi, Luis. I mean, great technology, amazing. Really, it's eye-opening. My question is more practical related. Do you have any occlusion balloons on the way that would allow a distal flow, something that was used at early PCI experience to maintain the flow? So this is one question. The other thing is a saline flash, as we do in OCT, not contrast, but saline flash with a quick pullback acquisition, of course, for retrospective analysis of the vessel, but this is something we currently use. So those two open questions, let's say.
Yes, absolutely. And that is something that we're doing the animals in the coronaries and in the other vessels. So the assumption is going to be very similar to OCT, the way this is being done. Yep.
Can you describe how far away you think this seeing through blood without balloon occlusion is? Do you see this in a 5-year plan? I'm a cardiologist in the San Francisco Bay Area, and really for the structural heart procedures like seeing the mitral valve, there's nothing that you can easily occlude proximally to be able to visualize these structures. You could saline irrigate for a microsecond, but obviously you would have to do it over and over and over again. And usually these patients would then develop heart failure just from the fluid. This idea of having no occlusion, where you could, in a chronic occluded vessel, you could occlude it proximally for a while and you'd still get away for the most part because it's already occluded. You're not making it much worse. But if you were really to expand it to structural heart, there's nothing you can occlude really proximally. Arrhythmia surgery, valve resections, or things like that. And to me, that idea of seeing through blood is really the holy grail of what is missing in all structural cardiac interventions. Can you comment on that, just the technology and where you think this is in 5 years? Are there a lot of other companies thinking about this? There's so much thought in this field about what you're just talking about, seeing through blood. People have thought about it for decades and have never really come up with anything that doesn't hemolyze or clot the blood around it. It's really, that's the science fiction, is this seeing through blood.
Yeah, absolutely. Yeah, thanks for the question. In a way, you have ultrasound that has intrinsic limitations about quality and about time resolution, but at the same time, you can see through blood by using sound. Everything that uses light has a major problem that the hemoglobin has a high coefficient extraction of light, meaning that hemoglobin is going to absorb all the light that goes through it. And that's the reason there has been this limitation for endoscopy to arrive into the cardiovascular field. What happened with this specific technology is that is scanning lasers. And as you know, there are some lasers that can go through tissue and blood. And those are the lasers that are used, for example, for OCT that has more penetration. So by using those particular lasers and coupling them with extremely high sensitive single photon detectors, which is basically you shine the laser through blood, some of that is going to go through, some that is going to come back, and then you are able to detect those photons coming back. You can reconstruct endoscopic images seen through blood, which is what you're seeing here, actually. So I think there is definitely a feasibility. Timing-wise, is going to depend on the amount of resources and basically you can allocate to push the technology forward. But that is one of the priorities of the team, is to making this reality, hopefully within the next 5 years. I'm not sure that Bahman is answer your question.
Yes, I think that's, to me, the most exciting part of what I'm seeing here is this ability where you don't have to occlude or irrigate constantly, but to do what the laparoscopic surgeon can do in the peritoneum with the Da Vinci robot. If you could do that endovascularly, you could just see what you need to see. You could see the surface. You said ultrasound, but forward-looking 4D ultrasound is not a particularly good technology. And a lot of the cardiac procedures, we use ultrasound, but you do a lot of pre-procedure planning with CT and MRI. You create some kind of a, you simulate what you're going to do, and all the technology just kind of springs into place. The valve just pops into place. It's not really surgery, in my opinion, what we are doing with structural interventions. It's either it deploys or it doesn't deploy. The laparoscopic surgeon that is trying to resect a tumor is actually doing surgery. They have 1,000 things under their fingers when they're in the abdomen, because they can insufflate with air or with saline. But inside a bloody chamber, you just can't see anything, and you can't do all the things that you could do, that you would want to do.
Mm-hmm. Yes, we completely agree with you. If you actually have an endoscope that can see through blood, you are going to be completely changing even the technologies that you have right now to treat valves or other problems. So
I would just add that the key priority for the research group is to get this resolution improved and get this version of the product out in the market as soon as possible. It will not be the first release, but most likely the second release. We're investing aggressively in, actually Patrick is helping us to clarify and improve the quality of the visions that you're seeing right now on the Seeing Through Blood laser. Anyway, good. Let's continue, Luis, and then we'll have more time for Q&A at the end.
Yeah, sounds good.
And by the way, Gerald just joined us. He's next to me. If somebody wants to ask questions on the business side later.
Perfect. Thank you, Juan. So, just to continue on the theme, we have been talking about dissections and thrombus, acute or subacute, and what happened when there is calcium. We always, we talk, at least in medical school, about this coronary calcium scoring, and then there are use of IVUS or OCT, trying to understand what type of calcium a patient have and when to use lithotripsy. And these are just some examples of what would you see with laser angioscopy. This, for example, is a nodule or nodular calcification that you can see that is highly dense calcium based on the micro-CT and very smooth in the surface, compared to this other lesion that is what we would call an eruptive nodule, which is this coral type calcification mixed with thrombus. And what was interesting, actually, and at least I haven't seen in my practice before, is this concept of having calcified nodules, but that are barely attached to the surface. Unfortunately, we have all done probably cases where when we are in the aortic arch and the arch is calcified, and then we have thromboembolic complications, and probably what we're knocking off are those basically nodules. And this is the other extreme when this is a calcified chronic total occlusion, that you can see that there is a highly calcified plaque, and then you can see that at the very, very bottom, there is a tiny channel or microchannel where we think that when we start the clinical experience, we could use actually to cross these lesions. What I've shown you so far are mostly stenotic lesions, but we can go to the other end about non-stenotic symptomatic lesions. you all know that cryptogenic stroke is very common. 1 out of 4 of strokes have no clear etiology. And we are still using narrowing 50% or more as a threshold to say that that plaque is symptomatic. And these are just examples showing that that may not be the case. This is a patient of my practice that had multiple strokes on the right side of the brain and extensive workup, including multiple CTs and angiograms. You can see that there is a calcified lesion, but no, the vessel is still pretty open and there was nothing truly concerning. But after 6-7 strokes on the right MCA, we decided to do an endarterectomy. And this is what you are seeing here. You can see that the external vessel looks pretty normal. And then when you look at the carotid bulb and the internal, you can see how he had multiple clots in the surface of the vessel that he was very likely releasing, causing these recurrent strokes over and over. And in this case, the vessel was minimally stenosed. And the same can happen with erosions. And this is a paper that we recently published in JACC. What you are seeing here is the surface of the vessel. And then I want you to notice these red areas. If you look with a scanning microscope, those areas actually look like this. Those are red blood cells that are covered or trapped by fibrin web, and they are forming because the endothelium has been peeled away. So what you're seeing here are true erosions, which is endothelium missing, and then the red blood cells get on the surface, and then you have a thin layer of fibrin, which is covering that. And the same happens with plaque fissures. You know, the problem of the fissure is like the vessel may be completely open, but there is a fracture in the surface. And you can see in this case that you could clearly see the fracture of the vessel with some clots bulging through, which corresponds to this. Following the theme of looking at the surface, this is a common problem that at least I face in my practice, which is a patient that had a recent stent placed. Could be weeks, could be a few months, and they are on dual antiplatelets, and they come to me with a brain bleed or with a major hemorrhage in the GI tract. And we're being asked if it's safe to stop dual antiplatelets. And they're basically asking us if the stent has endothelialized, if it's covered by intima. And it's really hard to tell right now. And this is the view that the laser angioscope will give you, which is you can see the surface of the vessel, and actually you are not seeing a stent, meaning that has been well covered by endothelium and intima, except for some focal parts, like you're seeing at the top over there, that you can still see the tines. And actually you can even see some clots at the level of the tines. And then you can see that there are other regions down there that are very well covered. And then there are other regions like here and there that the coverage is suboptimal, and you can see some small clots in the surface of the device. Well, I think we have just 5 minutes left. Stop here. We can open for more questions.
Yeah, this was fantastic. I'm sure most of the people attending have never seen videos like this, unless you are on LinkedIn, but you can see how dramatically this can improve the quality of a patient life. So happy to answer any questions. By the way, Gerald McMorrow, the CEO, founder of the company, is next to me. So if you want to ask either medical questions or business questions, we'll be happy to answer them. There were a couple of questions online, but I think I addressed them all so far. I don't think I missed anybody. So I'll leave it up to you all to ask questions. Lucas, you have one more question? You're muted, Lucas.
Yes, thank you. Thank you, Luis. That's really great and fantastic. Again, do you have any experience when looking at the stents to use this for peri-procedural guidance, especially in things like bifurcations two stent techniques where it's really critical to get through the right opening of the stent, not to get under the stents and then optimize. How well that could play? Do you have an experience on that? Because I think we spoke a lot about diagnostic part, but procedural guidance, this is where OCT and IVUS came in as class 1 in US and Europe now. So because of that classification down the road, yeah.
That is definitely something that we want to explore. You know, we have done procedures, but most of the procedures that I have done, I am looking for is things that would do an endovascular neurosurgeons, like, you know, coiling of the procedures or using stents to retrieve clots. One of the main things that we would like to do is to form a group of physicians that want to pioneer this technology in their, you know, respective areas. And no, definitely interventional cardiologies and use this for white stenting under direct visualization is one of the things that I think would be very exciting. So if there is interest, you know, please reach out to Juan, and then, you know, we can add you to the team.
We've completed the October trial, which was a OCT guided bifurcation, which really led the recent upgrade in the guidelines. And I think this is the most challenging type of stenting. We call it DK crash or the culotte and stuff, but this is why stenting, let's say, but exactly why I see the biggest value in terms of procedural guidance. So very, very, very nice that you think about things.
Absolutely.
Thank you, Lucas. Anyone else? Justin?
Can I ask a business question?
Sure.
Just about focus and timing, I suppose. So, I mean, we've seen a lot today. Congratulations, your technology looks, you know, really game changing. There's a lot of incremental improvements. This looks like something totally new and revolutionary. So congratulations for working on it. There's a lot here. You know, we've talked about the application of this in 3 different specialties, I think, you know, neuro intervention, interventional cardiology, peripheral intervention. And then obviously there's the geographic element of focus. You know, there's Americans on the call, there's Europeans on the call, you're going to do a call in Asia. PAC, help us understand a little bit I suppose the road, you know, where the company's at now. I hear you've just raised Series A. Help us understand a little bit where your focus is and, you know, how you eventually get this into folks like Lucas's hands. Thanks.
No, yeah, perfect question. Actually, that sets us up for explaining how we're going through the FDA process. We have already met with the FDA. They have given us their feedback as to what are the issues that they are concerned about. There's nothing in the pre-submittal letter that are significant blockages for us. So we know what the path forward is. The money that we raise is help us to achieve those milestones. We're going to go through what they call the breakthrough device designation.
Yep.
And that will allow us to speed up the development of the company and be able to get reimbursed and work with CMS on that. So those are our next steps. Yes, of course, we're going to be very focused in the US first. We're going to be very focused on a couple of applications like CTOs, because that's our beachhead, if you want to call it that. And from there, we'll grow. But as cash flow kicks in, we'll be able to expand into other applications. What Luis referred to is getting us a set of KOLs that are willing to work with us across, you know, head to toe. So we expect to have indications, you know, done by other clinicians besides our group. And we're hoping to collect between 20 and 40 KOLs and research groups that are going to help us with this effort. So in terms of timing and cash, we have the cash to get us to cash flow positive sometime in, you know, late 26. In terms of technology, the red, blue, green laser is the first one that will come out into the market. He didn't discuss the OCT version, but there's going to be an OCT version added to the red, blue, green later in 26. The research that we're funding for the see-through blood probably will take about 18 months. And then, of course, go through the FDA process. So that's probably early 27. So I think if we do our job right in terms of hiring the right support staff and consultants to get us through the FDA, we should have this product into the market in early 26, probably by Q2. We'll build our sales force here in the US first. And then with the FDA, we plan to do not just the US, but also Canada concurrently. And after that, we'll go to the UK and the EU for a CE mark. And you know, that takes time and effort because every country is a little different. And we hope to work with multiple distributors or partners for the rest of the world. There's no way we as a company can build a sales force for the rest of the world concurrently. So those are the steps we're going to take forward.
That summarizes it pretty well.
Justin, hopefully that gives you a sense as to where we are and what our go-to-market strategy is all about.
Yeah, sounds really logical. Thanks for the explanation. And then just in terms of the specialties, I guess that's the final one. Are you more cardiology, neurointervention, peripheral focused at the beginning?
Luis? Yeah, Luis, you want to expand?
Yeah, it's going to be a combination between peripheral and interventional cardiology.
Yep.
The expectation is going to be clear for a 510(k) path without clinical data. And one of the first things that we need to do is the procedural development, like understanding exactly how the technology is going to be used in the different vascular fields. And then based on that, then is to find indications and then construct prospective registries looking for basically specific indications for that particular condition. So, it's a few years process, but that's the.
Congratulations.
Thank you. Joel, you have a question?
Oh, well, I think actually Luis has just answered it at the very end of his talk about how things are going to move forward from a research perspective. I guess it's a little disappointing sitting here in Europe that your plan is North America first. Well, often getting a CE mark is actually easier than getting FDA approval a lot of the time. Getting reimbursement in Europe is very difficult, but getting the product so that it can be physically used in a clinical setting is not always the most challenging thing. We've certainly had very early technology with CE marks in our hospitals before. So I would hopefully you won't find that as challenging as perhaps it might appear from the other side of the pond.
Yeah, and you're right, Joel. It's just a matter of cash flow, right? I mean, if we are able to we hope to raise a Series B later this year for 50 million or so, right? And if we have enough money in the bank, we can hire the right team and expand our team to help us fast track some of this stuff. But as a small company, as you know, we're a startup, it would take time and effort. But Gerald has the whole idea to eventually grow to about 400 people at one point in the next 3 years. So we're going to be very aggressive with our path forward, but it's just a matter of managing the bottom line, right? So we're still a business. So we hope to be able to help you, provide this technology in Europe in probably late 26, if not earlier. It just depends on how quickly we can raise additional funding.
Yeah, just a comment about that. We talk about the resources and the regulatory path. I think what is critical as well, that this is a new technology and where is deployed early on is going to be as well depending on where is the team, where are the champions, the physicians that really want to pioneer and push this forward and basically learn and train the next generation of users. So
I agree. Yeah. I think a registry looking at, as Lucas said, looking at the help with procedural indications, particularly around bifurcation, stenting, DK crush, clot, seeing those indications and doing that'll be a really beautiful thing because you could see it. It would be so attractive for people to be able to see it, and it would have a wider impact on more physicians than a sort of focused complex CTO indication, where certainly in Europe that's a relatively small number of operators focus on that area, but a lot of people are still doing bifurcation PCI day in, day out, and would really benefit from optimizing their stent technology.
Yeah, I know there are more questions. Obviously, we're running out of time, but I wanted to make sure you understood the reason for this meeting today is to encourage questions like yours and to challenge us as to make sure that we are understanding where you think this technology needs to go. And the whole idea of having the early access program is to create a group of knowledgeable people that can guide us in our development. We think we know what we're doing, but it's always good to learn from others, especially people actually in practice, to help us identify the right path forward, even as basic as does the UI make sense? We have a user interface. Does it make sense for the practice? We have all those questions ourselves. And so we hope to create a user group, if you want to call it that can give us early access and early feedback. And then we'll provide, for academic research, the right tools for them to begin to do some of this early testing so that we can use that technology or those results to apply to the FDA for the 510(k). So that's kind of the plan. So you're going to hear from us after this event. We're going to send an email asking you for feedback on the event and asking you who may want to participate in these 3 categories. If you're an academic research group or you want to have access to the device for testing, and you can help us write some articles about it. And you have a chat with Luis once a month. And that's the user group that I'm working to establish with you all and have access to this information as early as possible. With that, Gerald, do you want to say anything?
Well, we hope to provide better imaging to allow you guys to see what you're doing in real time, and team's working away on that, and we look forward to working with you all.
Thanks, Gerald. Any other questions before we wrap this up?
Yes, would the Middle East be integrated in your commercial launch like Egypt?
It's one of those things. I'm actually having discussions with a group out of India, another group out of the UAE.
In your research before the commercial launch, before we would like to be also in the lead of the use of this technology, but I'm asking about would be you mentioned Europe as a target. Would you be interested in investing in the Middle East too for the research purpose, or what?
Yeah, I don't see why not. As I said to you, the rest of the world is also important to us. Australia, obviously India, Dubai and Saudi, those areas of the world are important for us to do some research. We were discussing the commercial approach, right? Because of the way we plan to go to market. But in terms of research, we are keen and open to discuss it with anybody in the world. So follow up with me after this call, and we'll discuss as to how you can get access to one of these tools when they become available. Fair?
Great.
Thank you.
Yeah. Anyone else?
Yeah. Yeah. So Juan, thanks for having me. I'll be interested in, because I've trained in intravascular imaging. I do a lot of OCT, IVUS. I'm quite interested in ACS research. I look forward to this technology and would be really interested if there's any scope for clinical research.
You sort of.
Am I audible?
Yeah. Can you
Absolutely. So the idea is to find basically physicians like peers. And they want to use this as a new tool to understand what's happening with our patient and how we can treat them better. So definitely, reach out to Juan.
Yeah, sure.
And then we can involve you.
And I had another question for you.
What, sorry?
I had another question for you. So you didn't mention about plaque erosion.
Yes.
Now in OCT, we with a fair specificity and sensitivity differentiate between a plaque rupture and erosion. As of today, this technology, how good it is to differentiate an erosion from a rupture?
Yes, I can show you. An image is worth 1,000 words here. So, you're probably familiar with this JACC paper about vulnerable or high-risk plaques.
Yeah.
This is how OCT shows a rupture, this ulcer here. This is how our scanner would look like. You're seeing the two ulcerations, excavations over there.
Mm-hmm.
This is how the erosion looks by OCT, which is hard to tell if it's actually an erosion or there is just a thick clot for some reason there. And this is how it would look like with angioscopy. And then finally, this is the third type of lesion, which is the eruptive calcified nodule. And this is how it would look like with OCT. Sorry, with the angioscopy. You can clearly see the different calcium phenotypes. This is a calcified sheath, benign, nothing really to do. This is the eruptive nodule, coral type of calcium mixed with thrombus. So
okay. But as of now, do we have any studies comparing? So comparing with either OCT or histology as a gold standard. Do we have any such studies?
We have done histology versus angioscopy, which is this, for example, the histology and angioscopy to see a fissure, but we haven't started the clinical experience yet.
Okay. So, thank you.
Mm-hmm.
Well, I hope you found this presentation fascinating. We obviously are very excited to bring this to the market as soon as possible. Feel free to send me an email or Coco or directly to Gerald. And we'll continue this conversation with you all, on a monthly basis. So Thank you so much for joining us. This presentation is being recorded, and it'll be on our website later on this week. And I hope to see most of the Europeans in Rotterdam when we're there next month. And you get to ask as many questions as you want. We'll have the device with us. You can see it in action. I think Luis is going to bring not just a device, but also we're bringing everything. Luis, are you bringing a Phantom?
Thank you. We can discuss offline.
Yeah. Okay. Good. Thank you all, and good to meet you all. Take care.
Bye, Juan. Thank you very much.
Thank you so much.